Hello, everyone, and welcome to today's webinar, Thinking Translational, Things to Consider in CAR T Cell Workflows. I am Sabrina Lemus of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more, visit miltenyibiotech.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Robert Davies, Cell Culture Product Manager at Miltenyi Biotec. Robert, you may now begin your presentation.
Hello, and welcome to today's webinar, Thinking Translational, Things to Consider in CAR T Workflows. I'm Robert Davies, Cell Culture Product Manager at Miltenyi Biotec. Before starting the presentation, please briefly acknowledge our disclaimer displayed on this slide here. Taking a look at today's agenda, we will first look at the path to cell therapies from early discovery to commercialization and the challenges that you might face in the different phases. Then, I will elaborate on the key points to consider when pursuing translation from small to large-scale manufacturing. Next, I will talk about the many suitable tools that enable a smooth transition from small-scale research to large-scale workflows. Finally, I will end the presentation with a brief summary and some suggested resources as a follow-up to this presentation. Let's start off with a question. What is your vision when it comes to CAR T cell therapy? Every one of us will have unique goals based on development timelines. However, I think we can all agree that our shared overall vision is to develop CAR T cells that work, translate them into the clinic, and successfully treat cancer patients. However, as we all know, this is a long process. It starts with basic research and discovery and goes into preclinical development, clinical studies, and ultimately results in a marketed therapy that can make a difference to a patient's life. Cell and gene therapies, including CAR T cells, come with a lot of unique challenges as a novel therapeutic approach due to its increased complexity as a living cell therapy. Similar to other therapies, development challenges differ from phase to phase. In a research setting, you need to narrow down targets, develop safe CAR T cells, and validate them. In the translational phase, you need to transfer your small-scale protocol to a large-scale process. Eventually, you will also need to address regulatory requirements, coordinate with an increasing number of enrolled patients, and keep an eye on costs. As your challenges change, so do your needs. At the discovery phase, you need a high degree of flexibility and a diverse set of methods, whereas later on, high standardization and process automation will be key. It is obvious that the path to a marketed CAR T cell therapy is not an easy one. However, success has been proven, as reflected by the increasing number of commercially available CAR T cell therapies. As of recently, there are 6 therapies on the market to treat CD19 and BCMA-positive malignancies. And a lot more are in the pipeline. A recent count shows a total of 1,115 clinical CAR T trials have been initiated. The number of clinical trials are expected to continue to increase, as shown by the trend line within the graph on the right-hand side. Miltenyi Biotec has been and continues to be very active in the field of cell and gene therapy. As such, we are familiar with its unique challenges and have developed integrated workflows solutions, and tools for the development of CAR T cell therapies. Given our experience and expertise in the CAR T research and manufacturing field, let's discuss some considerations that can help streamline the transition from small-scale research to large-scale manufacturing. When pursuing the path to translation, it helps to plan with the end in mind. Consideration towards reagents, devices, and methods ahead of time can help avoid added cost and keep your development timelines on track. We will use the following workflow depicted here as a way of illustrating the steps involved with the generation or manufacturing of CAR T cells. First, starting with human blood products, followed by T cell isolation, activation via CD3, CD28 receptor engagement, gene delivery, the ex vivo expansion, as well as phenotypic and functional verification. We integrated and developed a broad range of products that are used in both research and clinical settings that can allow for a seamless transition of your whole research workflow to the CliniMACS Prodigy platform. Automating an end-to-end closed system makes manufacturing more scalable, robust, and reliable. Human error is frequently identified as one of the highest risk elements of a manufacturing process, leading to protocol deviations and batch failures. Automation mitigates these risks by providing repeatable and reliable cell processing. In addition to having a scalable, robust, and reliable platform for manufacturing, the CliniMACS Prodigy platform features a specialized T-cell transduction process that is uniquely designed to support the needs of T-cell manufacturing, including CAR T cells, inclusive of all the critical steps from cell separation to final product formulation. While we may speak positively about our own products that support the development of CAR T therapies, don't just take our word for it. There have been several peer-reviewed studies published in the last several years that can attest to our success in supporting T-cell engineering. Now let's take a closer look at the workflow itself and the various solutions to consider to help transition the development process to commercialization. T-cell isolation is the first step we'll review. In a research setting, several methods are frequently used depending on the type of starting material and cell population targeted for isolation. When it comes to translation, there are some considerations, including optimal reproducibility of results, reagents available in GMP quality, isolation methods that can translate to an automated system, and clinical process that may already been established or exist. Miltenyi Biotec is well known for its cell separation technology, so I invite you to browse our website to learn more about this technology. You can always reach out to me to help you find the resources you need to help guide the transition of your cell separation process from research to clinical applications. The next step after T-cell separation is reliable cell activation to facilitate 1. Successful transduction, and 2. An efficient expansion of the gene-engineered T cells. There are several solutions available that can provide cell activation as listed here, but there are several considerations to keep in mind, such as optimal reproducibility of results, reagents available in GMP quality, activation reagents that are suitable for automated closed systems, clinical processes that may already have been established or exist, a proven track record, and safety. Given these considerations, especially for translational applications aimed towards CAR T cell needs in both the research and clinical setting. Miltenyi Biotec developed an innovative activation reagent called T-cell Transact. T-cell Transact activates T cells classically via CD3, CD28 engagement. However, as a polymeric nanomatrix, T-cell Transact can be easily dosed volumetrically, can be removed by simple washing, and is available in a GMP-compliant format. This not only facilitates more robust T-cell activation, but most importantly, eliminates tedious debeating. This product is fully compatible with automated manufacturing platforms such as the CliniMACS Prodigy, making the translatability of T-cell activation as easy as it gets. Shown here, you can see that 24 hours after the addition of T-cell Transact, we observed typical T-cell cluster formation indicative of T-cell activation. After 48 hours, T-cell activation was verified by flow cytometry. As shown, activation markers CD25 and CD69 were found to be upregulated. Up to 80% of the T-cell population expressed both markers. Now that we've seen the results, let's take a step back and briefly review the process that leads to successful cell activation using T-cell Transact. Here, the T cells were pelleted and resuspended at a concentration of 10 to the 6 cells per mil in CliniMACS medium supplemented with T-cell Transact and IL-7, IL-15 cytokines for activation. From this cell suspension, 2 mils were filled into a 24-well plate. 24 hours after seeding, the cells were transduced by adding the appropriate amount of lentiviral vectors. 3 days after activation medium was replaced by CliniMACS medium supplemented with IL-7, IL-15 cytokines. The transduced cells were split at least every third day or when the cell confluence exceeded 70%. In some cases, translatability can be easier said than done. As such, we've developed a resource that directly compares the performance of research use and GMP-compliant Transact using a method similar to the one shown in the previous slide. I invite you to read through this resource to gain a better understanding of how both research use and GMP-compliant T-cell Transact show identical results with strong reproducibility. I invite you to download this app note that has recently been published on our website to learn more about the data that supports this conclusion in further detail. There are many solutions on the market that can support cell expansion, compatible with either serum supplemented or serum-free media. However, there are several considerations to think about when planning ahead for translation, such as reproducibility of cell culture results, reagents being available in GMP quality, medium that's available with or without phenol red, bottles versus bags, and unit-specific dosing. When you eventually transition to clinical applications, using a serum-free workflow is advantageous. For clinical CAR T cell manufacturing, we developed CliniMACS medium to support serum-free, GMP-compliant, and highly reproducible cell expansion. Cytokines manufactured by Miltenyi Biotec feature lot-specific biological activity to even better ensure reproducible results, regardless of manual or automated processes. Lot-specific activity can help avoid oversaturation to better ensure the expansion of memory T cells, as opposed to effector T cells that are not as ideal for CAR Ts. As depicted here, various starting material, healthy donor, different patients, frozen samples, can be used with no effect on cell counts, expansion, and viability when following this workflow. Now let's switch gears and discuss cell analytics. In both research and clinical settings, there is a need to analyze results, with the most common method being flow cytometry. As we've done with the previous workflow steps, there are a few considerations to think about, including reproducibility of flow results, confirmed performance of the designed panel, harmonization of instruments, user dependency of analysis, reports, and protocols. Consistent with our ability to address the needs shared by both researchers and manufacturers alike, our approach to flow cytometry is centered partly on the aim towards reducing the variability affecting cell analytics. While our affinity antibodies provide high lot-to-lot consistency due to their unique design, their use with our MaxQuant instruments and corresponding software are all made to create reliable and reproducible flow analytics. With the SmartGain technology, you can take an instrument setting from one device to another and achieve equivalent performance. The idea is to provide reliable data from test to test, user to user, instrument to instrument, and across sites. With express modes, automated gating adjustments based on algorithms can avoid variations caused by human intervention. The gating can be preset, making the analysis automated, leaving the user with time to do other things. As an extension of these capabilities, we support a high degree of automation with customization options as needed as a part of our continual application support. Since we are discussing our support for CAR T therapy development, it's worth taking some time to talk about our CAR detection reagents. The CAR detection reagents are primarily comprised of our reaffinity antibodies, providing reliable lot-to-lot consistency and designed to detect engineered T cells expressing CD19 or CD20 CARs on the cell surface. The reagents come pre-titrated, ready to use, while also featuring a mutated IgG1 Fc to reduce background staining. When avoiding the need to manually dose antibodies and/or perform FCR blocking, altogether, these features allow for reduced manual intervention, leading to simplified assays and improved reproducibility. In addition to the CAR detection reagents, Miltenyi Biotec offers several reaffinity stain express flow panels that come as dried, pre-formulated antibody cocktails. The Stain Express panels are compatible with the automated express mode analysis software to standardize and greatly reduce hands-on time when performing analysis on your resulting CAR T cells. Standardization doesn't necessarily need to come at the expense of flexibility. As you can see, there are several predefined panels that can support the various development phases of CAR T cell therapy, from small-scale research to full-scale manufacturing. Now that we're at the home stretch, I'll quickly summarize this presentation and provide some suggested resources to review as follow-up. I hope it has become clear why thinking translational early on is beneficial to ease the transition from the research phase to commercial manufacturing. As we've demonstrated throughout this presentation, the workflow for T-cell engineering is complex and requires many specialized solutions. Keeping your development plans on time when making the transition from research to clinical application requires planning ahead to ensure that each step that was previously manual in a small scale setting can be supported by automated, easy to use, and GMP compliant solution that are ideal for commercial manufacturing. For nearly every research product mentioned in this workflow, a counterpart exists that can support GMP process. A few things before we end the presentation. As follow-up to this presentation, please consider reviewing the following resources by scanning the QR code provided. We hope that you'll gain a better understanding on how Miltenyi Biotec can support your CAR T-cell therapy development and other T-cell based applications. If you have any questions or want to discuss how Miltenyi Biotec can support your unique needs, Miltenyi Biotec is present in 23 countries, and we have representatives in a total of 73 countries worldwide. I also invite you to follow us on the various social media platforms, LinkedIn, Facebook, Instagram. Stay tuned and don't miss our latest scientific publications, free research protocols, travel awards, and latest product releases. If you have any specific questions to this webinar, please write us an email and we will get back to you as soon as possible. Thank you for your time and have a wonderful day.
Thank you, Robert, for your informative presentation. We are also joined by Robert's colleague, Lorenz Fulla, Global Product Manager at Miltenyi Biotec. We now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So our first question I have here for you guys: how does the straight form whole blood technology works?
Oh yeah, that's an interesting question. Just to reiterate a little bit on that. So straight from is actually rather simple. It works just like regular microbeads. However, the straight from whole blood microbeads actually enable a direct magnetic cell separation from whole blood, various whole blood products, for example, leukoparks or LRCs or others. And you can enrich various leukocyte subsets, CD4 positive, CD8 positive T cells, monocytes, so CD14 positive T cells, also B cells. And you can also do that either manually using our manual separators or also automated using our various automated instruments. And what else? The best thing is actually that you don't need to perform any density gradient centrifugation, but you can just directly use the isolated cells for your CAR T cell generation or for flow cytometric analysis or other, for example, also molecular biological applications or functional studies also.
Thank you for that answer. The next one I have: what's the difference between Transact and other bead-based simulation reagents?
Rob, do you want or should I?
Sure, I can take that. So with Transact, the important aspect I think with this, as opposed to other bead-based simulation, is that there is no debeading that really needs to be involved. It's volumetrically dosed, so you don't actually have to do any math in terms of cells to bead ratio. It's volumetrically dosed, so that is an easy to use part of the aspect of the product. But it's also not a bead, so it's actually no debeading is needed. So it actually, with a simple media wash, after activation, the dilution out of the Transact is all that's needed and sufficient for to remove Transact from your cell culture. So the other aspect of it is that it is a colloidal nanomatrix, meaning that there's really no vortexing involved. So the need for being able to do pre-mixing is greatly reduced. So altogether, it comes off as a really really nice, easy to use, off-the-shelf product for T cell stimulation.
Yeah, just to add on that, I mean, it's also suitable for the CliniMACS Prodigy, right? Because we have it also in Max GMP grade. I think that's also
right. That's exactly it, and that definitely is a huge support of our translatability aspect of our CAR T workflow, and that is with the cytokines for expansion, Transact for activation, as well as our TextMAX T cell media. Both exist in both RUO and GMP formats. So those three main pillars of T cell cultivation are fully translatable.
Awesome. And then it looks like we have one more question here. How do I know that the MaxQuant is a good fit for both clinical and research applications?
Do you want to start, Rob?
I think just to kind of start us off with that, I think the main aspect is really the automation that's ingrained within the Max Quant. Lends itself, to both research and clinical applications. So, if something is determined to be optimized in a research or even a process development setting, that's something that can actually be carried over quite easily within the instruments to a commercial setting that might be really more applicable or more important to IPC QC functionality. Lorenz,
just to add on that, Miltenyi has done a great job to really develop a whole family of Max Quant flow cytometers. So I think until now we actually have 4 different instruments really fitting to specific applications. So like you said, whether you're coming from research or clinical applications, you can just get the instrument that really is attuned to your specific applications, to your specific needs, be it high automation, high parameters, or also optimized for fluorescent protein detection, for example.
Right, exactly. And I really think the preset panels, the antibody cocktails, and the various software capabilities really lend itself to making it really very, very, very streamlined. Very minimal manual intervention is needed, and that can be definitely a benefit for both research and commercial applications.
Oh yeah, exactly. Nice.
Are there any other questions out
there? That looks like all the questions we have for today. Robert and Lorenz, do you have any final comments for our audience?
The only final comment I would have is, please don't be shy in reaching out to us, to explore, more about our CAR T solutions for both research and clinical. There's a lot more that we offer and you can learn about than what we covered in this presentation. There is obviously within CAR T, we are well established in being able to provide holistic workflows, starting material to, final finish. So I would invite anyone that is curious, that would like to try anything, or just wants to be able, or would like to have talk to a sales representative, please let us know. We can definitely start to help you guys, elevate your CAR T research and clinical applications.
Well said. I don't have much to add. Thank you very much.
Yep, I think so too. Thank you very much.
All right. Thank you so much, guys. Thank you again, Robert and Lorenz, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I would like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, or those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of your registration. And this webcast can also be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. And until next time, goodbye.